All of us are unfortunately painfully aware of the severity of the epidemic of C. difficile. So wouldn't it be great if there was a vaccine that could be used to prevent the onset of symptoms of C. difficile and to prevent the infection? And this is what Pfizer is currently working on in a phase 3 trial. But this week, in the clinical infectious disease, they presented their phase 2 data looking at a vaccine that they are developing. Their therapeutic target is an antibody response to toxin A and toxin B. And as we've discussed in other sections, the original ideas behind antibody responses to toxin A and toxin B came from studies by Kieran Kelly and Lorraine Kine in the late 1990s, where they showed that an immunoglobulin response to toxin A prevented 48-fold onset of C. difficile infection in individuals who received an antibiotic for other indications. And they showed also in individuals who had active C. difficile that the immunoglobulin response to toxin A also prevented recurrence. This became the therapeutic target for bezlotuximab, a fully humanized monoclonal antibody that has been shown in phase 3 randomized controlled trials to prevent recurrence and is now FDA approved as an accessory treatment, a treatment in addition to an antimicrobial, to be effective at reducing recurrence rates. But with this vaccine, the target is to give individuals who are at greatest risk for C. difficile infection the vaccine and prevent the infection in the first place by stimulating an immunologic response to toxin A and toxin B. Within this phase 2 trial, 855 patients were included. They were aged 65 to 85 years old, and it was conducted between 2015 and 2017. Patients were randomized to either receive placebo, a 100 microgram dose, or a 200 microgram dose in a 1:3:3 ratio. So the majority of the individuals did receive active vaccine at varying doses. Overall, there were 2 treatment regimens that were looked at. The days regimen, where patients were given vaccine or placebo at day 1, day 8, or day 30, versus the month format, where individuals were given vaccine at month 0, month 1, and at 6 months. And what we're looking at on this slide are the outcomes. Plainly stated, the days protocol, where the placebo or the vaccine was given at day 1, day 8, and day 30, was less effective than the month protocol. In the month protocol, responses were seen at month 0, month 1, and the booster at 6 months seemed to have a significant effect. Within these graphs, what they're showing is the percentage response of what is believed to prevent C. difficile infection. And we can see very nice immunogenic responses, with the peak response in the month protocol being at 7 months. Overall, when they looked at the safety of these treatments, they seem to be very, very safe, with the only adverse effect being injection site pain, which we see with most vaccines and most injections. So overall, this is a really exciting study because it's showing in phase 2 data that this vaccine that's being developed stimulates a good immunogenic response to toxin A and toxin B, and it's safe. And we are really excited to see when the phase 3 data comes out, which is looking at a much larger population and seeing and following them for an extended period of time to see whether this actually works and prevents C. difficile infection in a broad population. If it does, this could potentially be a large game changer for our patients at greatest risk for C. difficile that we could identify, give this injection, and then potentially prevent the infection in the first place.